After watching this video, you should be able to explain the principle of the Kjeldahl method for crude protein determination, identify the main steps involved in the Kjeldahl process, digestion, distillation and titration, recognize the reagents, catalyst and equipment required for the Kjeldahl analysis, describe the role of each stage, digestion, distillation and titration, converting organic nitrogen to measurable ammonium ions and to protein. Hello everyone, today I am going to demonstrate you about the Kjeldahl method, which we can use to determine total nitrogen or protein content. This is the reference method for determining the protein content in any material including food. Proteins have a major role by providing amino acids for the growth, repair and maintenance of the body tissues. And it is also an energy-giving nutrient in the diet. Each gram of protein provides about 4 kilocalories of energy, which requires for us in our day-to-day activities such as playing, gaming, flying and dancing. Therefore, providing the body with good quality proteins through its diet is very essential. Protein structure is organized into 4 levels. Primary structure, that is the sequence of amino acids. Secondary structure, local folding into alpha helix and beta pleated sheets. Tertiary structure, the overall 3D shape of a single polypeptide chain. And quaternary structure, the assembly of multiple polypeptide subunits. This folding, driven by various chemical interactions, determine the protein's unique function. Now let's see how do we measure the amount of protein in our food. Food protein is mainly quantified in terms of its nitrogen content. Assuming all nitrogen from proteins of that food, it's highly depend on the degree of sequence of amino acids. Then we convert the quantified nitrogen into protein using the conversion factor, obtain the crude protein content in a given food sample. It's possible to determine the total nitrogen content in samples such as foods, beverages, agricultural products, environmental samples, chemicals, biochemicals and pharmaceuticals. The protein content of organic matter can be determined mainly by 2 ways. Direct method, directly measured by specific chemicals or physical properties unique to proteins. Indirect method, indirectly by determining their nitrogen content, for example, Kjeldahl method, Doomer's method and combustion methods. Today we will take you through a widely used analytical technique called the Kjeldahl method, which is an indirect method to determine the total nitrogen content in various nitrogen containing substances. This method was introduced by John Kjeldahl in the year 1883. The Kjeldahl method involves 3 main steps. The first step is the digestion step. The second step is the distillation step. And the final step is the titration. The Kjeldahl method can be used to analyze the protein content of a wide range of samples, a tea leaf samples, any pharmaceutical products, any type of dairy product, any fruits and vegetables, and even any type of grains, and a soya meat sample as well. Now let's begin with the digestion step. So first of all, you have to do the sample preparation. So you have the soya meat sample where you wanted to get a homogenized soya meat sample. And next step, we need to measure accurately 1 gram of your sample and then place the weighing board inside this. Then press stir. You have measured accurately 1.080 grams of your solid food sample. Then you have to transfer your accurately weighed food sample into the Kjeldahl digestion tube. Then you have to add 25 milliliter of corn sulfuric into the digestion flask. Next you have to add 2 Kjeldahl catalyst tablets into the same digestion flask. Next you have to place the digestion tube in the block digester for the digestion process. This Kjeldahl instrument consists of 3 main units. The first unit is the digestion unit, where the digestion is taking place. So, which is connected with the scrubber unit. The purpose of having the scrubber unit is to neutralize all the harmful gases evolved from the digestion process. Similar way, you have to weigh accurately 5 different weights of your same solid food sample here, and a blank test is required for the Kjeldahl analysis, where you, without adding your sample, you have to add all the reagents into the blank experiment. Now place the digestion tubes in the block digester. Then connected the scrubber unit with the digestion tubes. And now you can start the digestion process. So you have to initially, at a slow rate, you have to increase the temperature gradually up to 400 Celsius. Then you have to switch on the digestion unit and then the scrubber unit. This is the distillation unit, where the distillation and neutralisation is happening. The receiving flask contains few drops of methyl red indicator. Then place the titration flask into the distillation unit. After the complete digestion process, you can observe a clear solution in which all the nitrogen in the sample has been converted into ammonium sulfate. The slight blue color can be observed in the digested sample due to the presence of copper sulfate as one of the components in the Kjeldahl catalyst tablets added during the digestion step. And now you are about to start the distillation process. To release ammonia, we first make the solution alkaline by adding a strong base, usually sodium hydroxide, to the Kjeldahl flask fixed here. This converts ammonium ion into ammonia gas. Boric acid will be automatically collected into the receiving flask containing the methyl red indicator, and liberated ammonia will be trapped in boric acid. Next, the mixture is heated. The released ammonia gas is distilled and carried by steam into a receiving flask. You can see the reaction appears on the screen here, where ammonia can be liberated as a product during the distillation step. The ammonia gas is what we want to measure as it directly relates to the protein content in your sample. So initially you observed the pink color in the titration flask because of the boric acid, the acidic color of the indicator methyl red. However, when the ammonia is liberated and coming through the condenser, that will be trapped inside the boric acid, and you observed the color change from pink to yellow, the basic color of the indicator. Now once the distillation process is completed, then you have to move to the titration step. Now let's move on to the final step of the Kjeldahl analysis, the titration step. So here this is the solution which we obtain after the distillation process. So similar way you have to carry out the rest of your solutions, the distillation process, and then do the titration step. Now we are about to start the titration process. So this is the auto zero burette where we are going to use for the titration. So you can observe here how the auto zero burette can make automatically the zero meniscus to the zero level. So you can observe it here. Now before starting the titration, you have to make sure are there any air bubbles trapped in between these tubes. And now it's about to start the titration. Now let's begin the titration. So here you have to add dropwise the standard HCl solution into the titration flask until you observe the color change from yellow to pink. Now you observe the color change from yellow to pink. Okay, now the color changed from yellow to pink. So now you can obtain the burette reading by looking at from the eye level. Okay. So with this burette reading, you can proceed with the calculation and you can determine the amount of nitrogen present in the soya meat sample we used here. And hence you can determine the total protein content present in your sample. That concludes our video on Kjeldahl method, where we have successfully converted organic nitrogen to ammonia and determined the crude protein content in a given sample. Thanks for watching this video, and we will meet you next time with another video.